Today we'll be looking at part 2 of biochemistry, once again using web pages developed by a teacher in Saratoga Springs, New York, Mrs. Jordy, and we appreciate the use of her hard work. Bio and chemistry, as we discussed earlier in the week, brings together two topics, biology and chemistry, and they go together. That's explains the chemistry of life, and there's a lot of chemistry in life. One thing this particular presentation is going to do is look at a very unique part of biochemistry called enzymes. Enzymes are a combination of form and fit. Form is enzymes can cause things to happen inside your body, cells and larger organs, and they also can bring things together. If you look on the screen there, lock and key theory, to make a key and a lock work, you have to put the key inside the lock, and the key has to have some kind of a physical arrangement on the end of it to make it hit the little tumblers inside there. Now, locks that use numbers and combinations are different, but old-fashioned keys really did have pieces of metal that were quite distinguishable. And if you look at a key to your house, it's probably got all sorts of little shaped triangles on the side of the key that goes into the door. If we want to make a bond between two biological things, could be the atoms or molecules, we can bring them together. This is a enzyme, this little creature thing here, and it's going to take that rectangle and this circle, and they fit precisely into this place on this molecule, and then they can be joined together. That would be forming a bond, and that would take some energy provided to the enzyme. We can also take an enzyme and put something into it, and we can break that thing apart, and we can get then the components of that bond, and we'll get energy out of this reaction. And enzymes also can speed up or slow down chemical reactions, generally speed up. And the enzymes are like a catalyst. They don't get used in the reaction. They don't get used up. They just can perform this function. Your parents' car probably has a catalytic converter on it, which helps with the pollution. And it has some materials that take the exhaust gases and make them into less harmful products. But the catalytic converter does not get used up in the process. Let's make sure we understand what a chemical reaction is. It's a process that changes one set of substances into another set. There's an iron device there. I'm not quite sure what that is over here. It looks like a wheel or something. And it's made of iron, and it's exposed to oxygen and water. And over time, we get rust. Now here up in central New York, we see a number of trucks driving around and other vehicles that have obviously been through a few winters up here with the heavy snow we get and a lot of salt on the roads, and they'll have rust around the fenders and doors and everything else. And rust is a natural product then of a reaction between iron and oxygen, and salt can help facilitate the reaction, so we can get a mess. How do you know a chemical reaction has taken place? Well, there'll be bubbling if you're doing it in a lab. There can be a spark. There can be a color change. There can be heat produced or used. The mixture can get warm or cold. We can smell a change, or hope it's not a dangerous substance. So it's generally easy to see if a chemical reaction has taken place. Rust takes place rather slowly, so we can't really observe that unless we really are bored and watching something rust, but you can see the result. Chemical equations, we take two products, or two things, A and B, and these are not products, these are what we call reactants. We gotta mix them together. And when we do that, we'll get something else called C, if these things can react. A series of chemical symbols and formulas to represent a chemical reaction. It's sort of like in math, where we use symbols to represent different things, variables and whatnot. So the reactants are the starting materials, and they react with each other. It's easy to remember. And the product is what we get, the end result. If there's a reaction, there may not be one. And the arrow shows how the reaction progresses. These two things form this. Here's some hydrogen peroxide, which is H2O2. It's water with extra oxygen in it, and it will break apart with an enzyme to be water and bubbly gas oxygen, and bubbles of oxygen will form. That's the reactant, that's the product. We put this stuff on cuts and scrapes because it oxidizes the skin where it was damaged, and that kills germs and bacteria in there. Have you ever put hydrogen peroxide on a cut? What happens when it comes in contact with the cut? Well, it kind of bubbles, and sometimes that scares you. Breakdown of peroxide into water and oxygen will occur very slowly, naturally, but if we put something in it to make it go faster, it can be kind of impressive. So here's the reaction. H2O2 is our reactant. It's only one thing. It's going to break down into water and oxygen. And these people are setting up. I found a YouTube that may or may not work because the internet's been slow this week, but let's give it a try. Let me turn the sound up a little bit. And it's got to try to find it. It may or may not. We'll see. This is exciting. Well, I may go to that reaction separately after the video. Let's give it another 10 seconds or so. We will stop that. And we'll look at that reaction later on. Let me copy the reaction here, or copy the link. And I think we just messed up the whole program. But we can start it again. Sorry for the technicality there. Let's see. That's not where I want to be. Let's try the right one here. Okay, that's pretty much where we were, and we'll look at that reaction later. This doesn't have it as a link here. Okay, we have to use potassium iodide, Ki, two particular elements together. Potassium iodide is the thing that makes this reaction work. In order to speed up reactions, we're adding something called a catalyst. I already mentioned the catalytic converter on your parents' car. Catalyst makes reaction work faster. And that stuff, if you go back one, we can see this tubes like this. When they made the reaction, it bubbled up rather nicely and gave them that. Looks like a big snowman. I don't know why they call it elephant's toothpaste. You can figure that one out for yourself. Okay, we have things called an enzyme. This would be a model of a catalase, which is a catalytic molecule inside your body, and the active site would be the middle of it, but it's got things around the edges that do stuff. The substrate is going to be the hydrogen peroxide. The substrate is what lands up on the enzyme. So we have the enzyme. It has a shape, sort of like a Pac-Man, and this thing has to be a very particular size to fit into that shape or it won't work. So enzymes are very particular as to what they'll work on and what they won't work on. So this stuff here will fit in there, and therefore it can be modified by the catalyst. Proteins are made up of amino acids that act as organic catalysts, and they speed up reactions. Reactant plus reactant equals the product. We know that already. And the enzyme is what makes it work faster. And in your body, we may want to be able to control the rate of a reaction. So nothing works without enzyme. I wouldn't say nothing, but all metabolic reactions in living organisms require some kind of an enzyme to work, which is kind of impressive. It's sort of like a control system for our bodies. And we have to have an enzyme to synthesize or build molecules, replace cells that have died. An enzyme would be what goes with this to make it go together. The yellow and the red can join in the presence of the enzyme. They might be able to do it without it, but at a much slower rate, and that wouldn't be satisfactory for the needs of your body. And sometimes you got to break down molecules or go the other way to digest them. So in this case, we take the yellow and the red. It's probably not the same stuff as up here, but we have to break it apart, and we get them separate products that we can use in some other way. They can be very specific little shapes. Now, these look like bolts and nuts and weird stuff, but in fact, some of these things are pretty weird. This would only fit in there. This is the enzyme that speeds up the reaction. The substrate is called the thing that goes in there. So it's sub, meaning it's less than this thing. And so this fits in there, and this can fit in here, and those are the only shapes that will work. So if there's other weird substrates floating around, they won't be able to work with this catalyst. These guys fit in here, and then they can get joined together, and that's what this catalyst is trying to do, produce this particular thing. I don't know what its function would be in your body or a body, but it has been formed quickly by this enzyme. How do they speed up reactions? Well, there's some picture things we can look at. If we have frogs that want to get into this water and they have to jump over this pile of rocks, they have to jump pretty high to get up there. That's called the activation energy, and not all the frogs will be able to do that. Some of them may not be able to jump high enough, and they'll never get to here. This area has a lower energy, and this one, the water level is lower, but we can make the enzyme make this wall lower, and then the frogs, except for this little guy, can get over and get to where they're trying to go. Enzymes help things happen in your body. So activation energy would be how much above this particular level right here do we have to go. It's almost be like having to push a car up a little hill before it could roll down a big hill on the other side. So without the activation energy, without the enzyme, I mean, we have to make this reaction go all the way up here to the top, and then the reaction can take place. But if we have an enzyme, we can only have to bring it up a little bit to activate it, and then back. Think of lighting a piece of paper on fire. If you're going to make a fire in the fireplace, you have to, the paper will burn, but you can't make the reaction take place until you start the paper burning. So the match that you light is the activation energy to start the fire on the paper that will hopefully make your fireplace work. So that's an activation energy, and you can worry about those little frogs if you want to. Okay, here's some other pictures of these. The active site, and then the substrates move into there, and they can be formed together, and we'll get a product. I have no idea what this is. It might be some kind of a molecule that has a very specific function in your brain or your liver or your stomach or your foot. No idea. Enzymes have a complex three-dimensional surface to which particular reactants called substrates tend to fit like a hand in a glove. An enzyme and a substrate bind tightly together, forming an enzyme substrate complex. The binding energy brings key atoms near each other and stresses the key covalent bonds, so it makes the bonds of these two guys be able to stick together and make a new product. As a result, a chemical reaction occurs within the active site, which is this stuff, forming a new product. The product then diffuses, goes away, freeing the enzyme to work again. The enzyme does not get changed in this process. There are millions of these processes going on inside your body right now, 24/7. You don't control it, you don't feel it, you don't even know about it, except from the results. And we get these weird pictures. So this is a three-dimensional object, a protein, and the active site is sort of like where this thing fits, and the substrate is what goes in there. Each enzyme has a specific characteristic shape that matches only one substrate. So think of that lock and key model. And we have names for these. Enzymes have the word ASE, like ACE, at the end. So sucrase breaks down sucrose, which is sugar. Protease breaks down protein. So these are kind of give it away. Sucrose, protein, lipid, and then ame is kind of a weirdo one there. Lipolase breaks down lipids, and amylase breaks down starch. So Here's an example of sucrose being broken down. Glucose and fructose, two sugars, can be separated by an enzyme into the two separate sugars, which might be different and have different uses in your body. And they're trying to give you little pictures here, but it's kind of confusing. But if you see something ending in ASE, it means it's an enzyme. That's a good general statement. And we get chemical reactions, and the enzyme molecule and the reactant molecule come together, and here they're breaking this one apart into two product molecules, the letter E almost, it looks like. Enzymes are not changed by the reaction, but the substrate is. These guys then are changed. This one here is broken apart, and these can go on their way. Enzymes can be reused again and again. It's the third time I've said that, but it's really important to understand that. Okay, here's starch breaking down glucose. Starch can be broken down into glucose, which is more usable for your body. But if you eat a ton of spaghetti tonight for dinner, some of it, if you don't need the energy contained, might land up as fat. So be careful, because glucose can then turn into lipids, which are fat. Proteins can be turned into amino acids, which are more useful for your body, and fat can be turned into fatty acids, which are useful, and glycerol. So these things are needed for your body to make different products that it needs to keep healthy. And these occur in your digestive system and your bloodstream throughout your body. Okay, and then you could say in this picture, what is identify X, Y, and Z? Well, X and Y are the substrate, and Z is the enzyme. And these guys are forming a starch molecule, which is then going to be broken up by this enzyme. Water is used in this one too. Why can molecule Z work on starch but not protein? Because protein doesn't have that particular shape. It won't fit onto the enzyme. Okay, and then back to our avocado from the lesson we had last time. Avocado has an enzyme that works on it, an enzyme prophenol oxidase, and there's that ASE word on the end that tells me it's an enzyme, in avocados causes the flesh to turn an unappealing brown color when exposed to oxygen. This process is known as oxidation. Oxidation occurs in lots of things. And how do environmental factors affect enzyme activity? Well, here's a substrate entering this. Sometimes it has to change shape just a little bit when that happens, and then we get this thing as being broken apart from these two green ones are the part of one complex molecule. They're being broken apart into two, and then they leave. How about frying an egg? You've all fried an egg, and you see that the egg white turns into, or the clear egg white turns into opaque white stuff, and obviously there's some kind of chemical reaction taking place there, but it's kind of hard to understand. And after the egg cooks a little bit, the white stuff won't run anymore. There's the egg turning into a fried egg. An egg's protein structure changes with the heat in the pan. The pan, it's because you're cooking it. The protein structure to start out with is tangled like this, but fluid. It can move as a liquid. It gets denatured or broken down in the heat, and the heat is what makes this enzyme work. The enzyme denatures or changes shape due to an increase in temperature, and the enzyme no longer works above 40 degrees centigrade, which is about 100 something degrees Fahrenheit. So the enzyme's not able to keep this thing together, and we have a functional protein when it's all stringy like this, and it becomes loose and like just a piece of thread on the floor because of the temperature or acidity change, but mostly temperature, and that's going to then make it so it increases the temperature and the pH changes the enzyme shape. Enzyme no longer has the correct specific shape, so it does not function properly and it can't flow anymore, and it tends to stick together. And that's why the white of the egg is a solid, whereas the clear part is liquid. All right, and we've also learned about pH or acidity. pH stands for potential hydrogen in chemistry, and pH things are basic, which is high pHs, and acidic, which is low pHs. Battery acid and hydrochloric acid are 0 and 1. Lemon juice is 2 and 3. Orange juice is about 4. Acid rain, 5. Black coffee, not good for you, 6. Urine is 6. Pure water is 7, right in the middle of that scale. Seawater, a little bit salty, so it has a slightly higher pH. Baking soda, 9. Ammonium solution, 10 and 11. Soapy water, about 12 and 13. And drain cleaner and oven cleaner are really, really basic, and they dissolve organic material. That's why if you have an oven full of things that are spilled over, then the high pH of an oven cleaner can break those things down. It can also happen to your skin or your eyes. That's why you don't want to get that stuff anywhere near your face. Okay, what happens here is we have a lot of hydrogen plus ions, which means they've lost their electron, and here we have a low number of those things, so it's just the same scale turned sideways. And all substances have a pH value, concentration of hydrogen ions in the solution, 0 to 14. 0 to 6 is acid, 7 is neutral, 8 to 14 is more. Environmental factors on enzyme activity. Enzymes work at particular temperature ranges. So if there's a particular enzyme here, it seems to work best at around 30 degrees centigrade, which is about 90 something Fahrenheit. And the rate of enzyme action here occurs at different pH. So temperature and pH can affect how an enzyme works. Very narrow range. If we wanted this thing to be maximum, we'd want 30 degrees centigrade and about pH of about 9. And that was where that enzyme would work best. If you did want that enzyme to work, you can make the pH low, make it acidic, and it wouldn't even work at all. So making this environment acidic would prevent this enzyme activity from taking place. And your body can use that to control how enzymes work. Optimum temperature and pH activity is the highest point on the graph. That's what we just did. And above the optimum, then they say that they change, that they denature, they break shape, change into another shape, or just become scattered little pieces. And we're not going to do this right now. We're going to do a lab next week where we're going to design an experiment that you can do at home. Labs are kind of hard in an online course because I can't ask you to mix a bunch of chemicals together and blow your kitchen up. That would not work well. But we'll be doing an experiment next week to understand how you can modify how an enzyme works. So that's the end of what we're going to do today. There's a couple of papers I want you to look at today. If you look at the website when you get home, we're already home. And if you have questions about how to turn those in, you can send me an email or call one of your friends or whatever. I'll stop there. Have a good day.